Module 13, the digestive system. The small intestine. Now it's time to discuss what happens to the chyme when it moves out of the stomach through the pyloric sphincter. At this point, it's in the small intestine. This is where most of the digestion of food takes place. You may remember that in the mouth, some starch digestion occurs when the food is mixed with saliva, and in the stomach, some protein digestion happens. But most food digestion occurs in the small intestine, and this is also where most of the absorption of nutrients into the bloodstream happens. Basically, the small intestine is where the chyme is further mixed and moved through the digestive tract until it reaches the large intestine. It's where food is digested and where nutrients are absorbed. Because of its digestive and absorptive responsibilities, the small intestine needs lots of surface area. It's quite long, too, extending about 20 feet in length. We can divide the small intestine into three major regions. The first is the duodenum. It's a smaller section, about a foot in length. In fact, the word duodenum literally means 2 and 10, referring to its 12 inches in length. The second region is called the jejunum, which is about 8 feet long. And the last region is the ileum, the longest region, which is about 11 feet long. The ileum connects with the large intestine. As I mentioned in an earlier section, the small intestine has the same layers of tissue as the stomach, with the exception of the missing oblique muscularis. And the duodenum has adventitia instead of the serosa layer. The great length is one way to provide lots of surface area for absorption in the small intestine. You see, its length gives the chyme a longer time to be in contact with the small intestine walls, so nutrients have a greater chance to move into the bloodstream. Additionally, there are rings of mucosa called circular folds, adding more surface area within that same 20-foot distance. And those folds have further extensions called intestinal villi to greatly increase the surface area. Intestinal villi are very small, like tiny fibers extending out of the mucosa. The free surface of the villi is made up of simple columnar epithelium. Notice that a blood capillary and a lymph vessel are found inside each villus. The lymph vessel is given a special name here. It's called a lacteal. So nutrients are absorbed through the simple columnar epithelium and move into the capillaries or lacteals so they can be distributed throughout the body. If you think the circular folds and intestinal villi are enough to provide adequate surface area, you would be wrong. The epithelial cells of the villi are covered with tinier microvilli that are extensions of the extensions. This helps to increase the surface area of the cells of the intestinal villi, so the nutrient absorption is even more efficient. If it weren't for the circular folds, the intestinal villi, and the microvilli, the small intestine would have to be up to 2¼ miles in length to have the same amount of surface area for absorption. Can you imagine how large our guts would have to be to accommodate that? Now let's talk about why there are capillaries and lacteals in the villi. First, blood capillaries are able to absorb the water-soluble nutrients, but they cannot take up fatty acids, which are the building blocks of fats. So the lacteals take in the fat-soluble nutrients and transport them into the blood by first making them a part of the lymph. Remember that lymph eventually makes it back to the bloodstream, so when it does, the fat-soluble nutrients enter the blood as well. Now the area inside the small intestine is referred to as the lumen. Lumen technically refers to the space in the center of a tube. So in the small intestine, the chyme travels through the lumen. In between the intestinal villi are special intestinal glands that secrete many materials into the lumen. In fact, there are both endocrine and exocrine glands there. Exocrine glands secrete materials through ducts. Mucus goblet cells are the one type of exocrine gland that secrete mucus. The mucus helps to protect the intestinal tissue from the chyme and from the digestive enzymes that break down the proteins, fats, and carbohydrates in the chyme. Remember, the cells of the intestine are made up of these same materials, so the body needs to digest the chyme without digesting the canal that carries it. Thus, mucus goblet cells are found throughout the small intestine. Some exocrine glands secrete fluid and enzymes into the intestine as well. In the duodenum, there are duodenal glands. Their job is to secrete a mucus that is alkaline in pH. Remember, the pH of the chyme in the stomach is very acidic due to the hydrochloric acid that was mixed with the food. As the acidic chyme enters the duodenum, it is necessary to neutralize its acidity, or else the small intestine would be injured. So the alkaline mucus does that. In fact, it is one of 4 secretions that are specifically designed to neutralize the acid in chyme. There are also endocrine glands here that secrete a hormone called secretin. This hormone isn't secreted into the lumen of the duodenum, though. Instead, it's secreted into the blood capillaries inside the duodenum wall. This hormone is controlled by the acidity of chyme. The more acidic the chyme is, the more secretin is released. You see, secretin inhibits the secretion of acidic gastric juice in the stomach, and it increases the secretions of the materials that reduce the acidity of chyme, also slowing the movement of the stomach muscles so the stomach empties into the duodenum more slowly. This is a perfect control mechanism to make sure the food entering the small intestine is not too acidic. A hormone that is secreted by the endocrine glands of the small intestine is called cholecystokinin. It's secreted as a response to chyme that has lots of fats in it. Chole literally means bile, cystic means sac, and kinin refers to contraction. So this hormone is responsible for the contraction of the bile sac, or gallbladder. Remember, bile helps to digest fats into fatty acids, so this hormone causes the gallbladder to contract, releasing its stored bile into the small intestine. Additionally, it increases the release of digestive enzymes by the pancreas, which also helps in fat digestion. Finally, this hormone slows the stomach muscles so it empties into the intestine more slowly. That's a good thing because it takes a longer time to digest fats, so this hormone works better to orchestrate fat digestion. A moment ago, I said that intestinal glands secrete fluids. Well, they secrete a lot of fluid. In fact, they can release up to 2 liters of fluid into the intestine each day. The juice that is secreted is alkaline in pH, so this is helpful to neutralize the acidic chyme. Additionally, the intestinal glands secrete lots of digestive enzymes. One major digestive enzyme is maltase. Remember, most enzymes' names end in ase, and that first part of the name refers to the chemical the enzyme breaks down. So maltase digests maltose, which is a sugar. Now, sugars are carbohydrates that can be monosaccharides, disaccharides, or polysaccharides. Maltose is a disaccharide, being composed of 2 glucose molecules. When maltase acts on maltose, it breaks up into its 2 glucose monosaccharides, which are then small enough to be absorbed through the intestinal villi. Another enzyme, sucrase, breaks down the sugar sucrose. Sucrose, or table sugar, is a disaccharide made of a glucose and a fructose molecule. Lactase breaks down lactose, yet another disaccharide. Lactose is a milk sugar made up of a glucose and a galactose molecule. You may have heard of some folks who are lactose intolerant. That means they have difficulty digesting the sugar in milk called lactose. This is because they cannot make lactase. The lactose sugar then moves through the intestine, causing discomfort or diarrhea. Most lactose intolerant people were once able to digest lactose when they were young, digesting that milk sugar with lactase as they were fed mother's milk. But lactase is an inducible enzyme, so if that enzyme is continually used, for example, by us regularly taking in milk and milk products, then we will continue to make it. But if we stop drinking milk, our bodies may stop producing lactase. Peptidase is an enzyme that breaks down peptides. Remember when I talked about the stomach that pepsinogen is secreted there, it's converted to pepsin, which breaks proteins down into peptides. Well, peptides are not small enough to be absorbed, so they have to be further broken down into amino acids. Then amino acids can be taken into the epithelial cells of the intestinal villi. Now think about that. The proteins that are ingested are broken down into their building blocks. Then those building blocks are brought to the cells of the body, which can rearrange them into the proteins that they need to make. Another enzyme I want to mention is enterokinase. It's not for breaking down food particles, but instead activates trypsinogen into trypsin. Trypsinogen is secreted by the pancreas, but it cannot do anything until it's converted into trypsin, when it will then break down proteins. This is kind of like the mechanism in the stomach where pepsinogen had to be converted to pepsin before it can break down proteins. You see, the cells of the pancreas are made primarily of proteins, so it would not be a good idea if the pancreas secreted a material that broke proteins down. So it secretes a precursor that when it is activated to trypsin in the small intestine will then do its job. So the small intestine is the major place where digestion and absorption occurs. Thus, it needs to be able to mix its contents around so that all the food material will be exposed to the enzymes and then will be exposed to the villi for absorption. Thus, the small intestine, like the stomach, also has motor functions. It can contract and relax so the contents inside get squished and pushed around. And like the esophagus, the small intestine has peristaltic waves, though they are weak ones, in order to push the food through the lumen.